Good afternoon. This is our Innovation Grand Rounds, opportunity for us to come together as a community to kind of celebrate medical innovation and ideas in our community with local role models and leaders. And today we have a unique opportunity where we have two individuals, a team of individuals that have worked together that really highlight the approach of identifying clinical problems and then finding technological solutions that can really address those. So I'd like to briefly just introduce both of our speakers today. The first, Graham Huesmann, he received his MD PhD in neuroscience here from our Medical Scholars Program and went on to do a neurology residency and epilepsy fellowship at Harvard Medical School and MGH, and then came back here to Carl and the University of Illinois, forming an epilepsy center. He's going to present on the second half, but talking about some of the clinical problems and applications. Our first speaker of this tag team is Professor Brad Sutton, who is also from the University of Illinois, received his bachelor's here in engineering, did his PhD at Michigan in biomedical engineering, and then came back here as is now a professor in our bioengineering department, as well as the technical director of the Beckman Biomedical Imaging Center here on campus. So I think it, again, is really timely. We'll also hear about some of the investments that Carl and campus has made collaboratively in the area of MRI. And it's a really exciting time, I think, for this technology and the advances, and really excited to have both of you here to highlight these. So thank you again, and I'll turn that over to Brad.
Great, thank you very much. So hopefully everyone can hear me okay. So we're going to show you what Graham and I have been working on over the last couple of years, maybe 4 years or so, and show you a new technique that can give different information in imaging with MRI, and then we'll show you some of the applications of it too. Normally you would maybe but the clinical drivers up front and then the engineering, but Graham is much more entertaining than me, so I'm going to go first. So I wanted to start by talking a little bit about how exciting MRI is in town lately too, just to give you guys a sense of kind of what's on the horizon. And if you heard Steve and I talking at the beginning, you heard some of these things already. But considering where we were 10 years ago and looking at where we're at now and where we're going to be in the next couple of months, it is so amazing. So 3T is the state-of-the-art magnetic field strength for MRI in clinical and in research use too. We have now three state-of-the-art 3T Prisma scanners, which Prisma is sort of the Cadillac of the 3T scanners. We also have two 3T Skyras in town now too. And then we just had a 9.4T Bruker preclinical system delivered. And so in terms of having really state-of-the-art equipment to do solid research and clinical work on, most of these systems are brand new in the last 2 to 3 years. And so it's really exciting to make that even better. We have a 7T Siemens Terra system coming September 10th. We won't actually get to start using that until December, so it takes a while for the installation to happen. But 7T is now becoming a clinical tool in addition to being a research tool. And so there's about a dozen of these in clinical centers across the US. And so we're really happy to be on the front end of this and partnering with Carl. It's a 50-50 between Carl and the university. So really a true partnership in order to bring that equipment to town. And then lots of other exciting things happening to go with this. Campus focusing on health sciences. Carl is making a Carl in Illinois, partnering together with this Terra to make the Carl Illinois Advanced Imaging Center. And then Carl is really coming up with an imaging center of excellence where they're getting faculty that have a research focus in that are really looking to push clinical research on the state-of-the-art imaging equipment there. And so really exciting time. It builds on our unmatched history. So I wanted to point out one other thing that's really exciting happening in the next couple of years too, and hopefully you'll be able to take part in this. But BIC, the Biomedical Imaging Center, where I'm a technical director, traces its roots to Paul Lauterber's lab. And Paul Lauterber won the Nobel Prize in 2003 for the invention of MRI. He was a professor at UC from 85 to 2007. He invented MRI at Stony Brook, but then came here in 85. He shared the Nobel Prize with Peter Mansfield. What people don't know is the role that Illinois played in all this development. So people know Paul Lauterber was here, but Peter Mansfield, who shared the Nobel Prize for the invention of MRI, was a postdoc here at Illinois. in the 60s. And if you look at all the commercialization, folks at GE that commercialized the first scanner, a lot of them were from Illinois. So we're really excited to celebrate that. It turns out that Paul Lauterbur invented MRI September 2nd, 1971. So 50 years from there is next year, September 2nd, 2021. And so we're planning on celebrating that. Last summer, or 2 summers ago, we found some artifacts in a shed. One of those was the data notebook from his original experiments back in 1971. And we also found 2 of his human MRI systems. So this just kind of shows you, this is the Nature paper that was published in '73, establishing MRI. And what I found in the shed was, you know, I would say never before seen, but I'm sure people in the '70s saw it. But this data slide was kind of lost, but we found his original lab notebook from back then. This is October 10th, 1971, the day in his invention statements that he said the MRI finally worked. So this is probably the first MRI data set. And you can see it goes directly with this 2-tube Phantom that he eventually published in Nature. So we're excited to do that. We also found his first 2 human MRI systems, 1975 and 1976. And we've been putting those back on display. It's great. We have this picture of Paul sitting here. This is called Big Red, and you can see why. It's red, and he has a person in the magnet. And so we've been able to find that. The console he's sitting at, the RF amp sitting up on top of here, and even the power cabinet for the magnet itself. And we've been able to put all those things back together. This exhibit will be open, you know, depending on COVID issues, will be open, and you can walk through. It's in the auditorium, just off the auditorium of Beckman Institute. And so if you're required to get twice weekly testing on campus, please come to the Beckman test site and look in the door while you're waiting in line. So it's right there in the line to get tested. So we're hoping to have a big party September 1st and 2nd to celebrate this MRI turning 50 years old. And we hope that people can travel again at that time and we can really celebrate this. So we're going to focus on this talk, as Steve said, showing you how we've developed some technology in MRI, and we were interested in looking at a few things associated with it, but then we really started working with Graham and finding that there are real clinical problems that can benefit from understanding the mechanical properties of the brain. And so from that point, then we were able to focus our development efforts on improving the resolution and signal that we were getting out of these to really target the applications to give Graham information about epilepsy and the hippocampus. So we hope from this you'll understand a little bit about this new imaging method, what's possible and how we can see that with MRI, what these elastograms mean, and then really to understand how that new information can be used clinically to improve outcomes. It's a big team required to do this, and so I'd like to point this out early on in the process too. A lot of people here locally, both at the university and at Carl, that have been really working on this and contributing over the years, but then also people at other places too. So we rely on some inversion algorithms from Dartmouth and other people that have contributed interesting components to this project. We require a lot of pieces to work together in order to get the images we get. So I'm just going to tell you what it is we're trying to measure and how we measure it, and try to give you a little bit of insight into why it's a challenging thing to measure, and then we'll let Graham tell you what some of the impacts are. So why would we, I want to start with this, why would we even measure mechanical properties in the brain? When we think about the brain, we think about, you know, the orientations of tissues, we think about axons, we think about all diffusion, and we think about lesions, but maybe mechanical properties wouldn't be the first thing to come to mind. And it turns out that mechanical properties and thinking about cellular biomechanics and extracellular matrix and the interactions between cells, once you start thinking about it, you realize there's a lot of information, a lot of potential information there about the brain. And so how do axons pack together? What is the organization like? How complex is that interaction between axons passing through tissue? What happens in inflammation? What happens with synaptic density changes and the like? And it turns out that mechanics is a really interesting modality everywhere in the body anyway, because if we start looking at the range, the orders of magnitude, the range of signals that we might get out of the body with something like X-ray CT or even some relaxation contrast with MRI, we see that we're limited in sort of the degrees or the magnitude of the change, how much of a difference you might have going from one tissue to another in the body. But when we start looking at mechanics, we have a huge order of magnitude, several orders of magnitude range of signals potentially to get, going from fat all the way up to bone And so this provides this nice wide range of signals. And so through that, we're hopeful that we can actually start to separate tissues based on a variety of the properties. There's also been some literature before we started this looking at decreases in stiffness and the like with neurodegeneration. And then there's a bunch of peripheral things like traumatic brain injury. How does your brain react to being impacted, and how do the mechanics of the brain interact with that? So there's a lot of peripheral things that were interesting too. So how might you measure the mechanical properties of the brain? I like to show this because this is where my group actually started. We were doing spin tagging, which is you can induce lines in the image with MRI. And we were pulling a little pin and dropping people's heads in the MRI scanner. And so let me see if I can get this movie to play. There we go. And you can see the head fall. It was only 2 centimeters. Yes, this was IRB approved, no problem. But you can actually see, if you watch the lines, you can actually see some of the movement of the tissue in here. It can be a very quantitative technique. You can actually, with the grid tagging, you can get quantitative information about the displacements of those lines when the head falls and hits, and you can understand, by looking at the gradients in those displacements, you can understand the strains in the tissues as you go through. So that's where we started, and we had to drop people's heads quite a few times in order to make those measurements. It's not a single shot thing. And so we're very happy that nowadays there's a new technique that can be used in order to image this information. And it's come into a field called magnetic resonance elastography, and we'll just call it MRE here. And MRE was first really used in the liver. This technology has been driven by Dick Eman at Mayo Clinic, and they've been using it in liver, and now they have replaced liver biopsies for staging liver disease with this MRE technique, where they can get really interesting information about fibrosis and different fibrosis levels in the liver, where with MRE you can sample the whole liver. You can get nice maps of where the stiffnesses are. Instead of, if you were doing biopsy, you would be sampling particular regions, and you could have a sampling issue that you may not sample all the regions that were the highest stiffness. You might miss them. And so with MRE, we get maps of these stiffnesses. And so basically MRE, we call it quantitative palpation. You can basically feel how stiff a structure is and make an image of that. So it's very specific, it's localizable, repeatable, non-invasive, a fairly comfortable experiment. There's this pad that sits on the surface, and it provides some vibrations. Those vibrations pass through the tissue. And then with the specialized MRI pulse sequence, we can sensitize ourselves to those displacements and pick up even the very small displacements of that in our images. So this is what it might look like. This is a gel phantom, and it has a stiffer gel in the middle and a less stiff gel on the outside. And we have our shaker device up here, which is called a resoundant, which puts pressure waves out. And we can watch those shear waves propagate across the tissue with MRI by encoding the displacements. And then we can put this into an inversion algorithm that basically does a finite element simulation, and we can estimate the material properties of the tissue from that. And so if we do that, we get this stiffness image here. Okay, so we require a couple things. We require an actuator to go with the MRI scanner, so an additional piece of equipment. And then we have to design specialized sequences that go with that, and that's what my group does. And then we have to have an inversion algorithm once we get these displacement waves out in order to get the material properties. And that's what we collaborate with people at Dartmouth for. And Aaron Anderson is our local expert here, who's a Beckman-Carl postdoc fellow. He's our local expert here on the mechanical inversion parts of this and the computation that goes with that. And he's on here too. So if we have questions about that later, he can answer them. So here's what it looks like for a person. We put a person in the scanner, and there's a pillow that goes behind their head. And there's this tube that connects to the resounding device, which sends the pressure waves out. And then we provide a pulse sequence that slides through. It provides some sensitivity, some sensitizing gradients here to the displacements that the head will be undergoing under those vibrations, which are about 50 hertz. And we can encode then even very small displacements. So with MRI, we're never going to see things below 1 millimeter or 0.5 millimeter with direct imaging. But here, because we're sensitizing the scan to the displacements with these encoding gradients, we're actually able to see a few microns of displacement in our images. And so we run a 1.6 millimeter imaging resolution coverage of the full brain in about 6 minutes. And the brain displacements that we're measuring are really about plus or minus 5 microns for the range. Pretty comfortable experiment. So about a 6-minute scan with this vibrating pillow under your head So not too bad. This is what the displacements in the brain might look like. And so you can see here from the scale, plus or minus 5 microns. We have to encode displacements in all 3 directions. So this is the head-foot direction, going up and down, and then the anterior-posterior, having your head move forward and backwards, and then left-right motion, we encode too. Even though we don't expect much left-right motion, we have a pillow behind people's heads, which is causing some nodding motion. So we get a lot of head-foot and anterior-posterior, but not too much left-right. And if you take those images and you invert them, you get some interesting information. And I'm going to tell you a little bit about what that information is here in the next couple of slides. So this is just an axial slice of the brain, just showing a T1 image. So you can see the gray matter and the white matter. And then I've outlined in red here the corpus callosum, and in blue the corona radiata. So corpus callosum are the fibers connecting brain right and left. Corona radiata are fibers that are heading up and down your head. And so they're both large fiber bundles. And so when we look at the stiffness map here, we're going to get 2 measures out. We're going to get a storage modulus and a loss modulus. So a stiffness and a loss component. And both sets of fibers are very stiff. They're very large. But then it turns out that the corpus callosum has a very different loss modulus than the corona radiata. And we'll tell you a little bit about that. And they also include here diffusion tensor imaging, which we can get with MRI. Diffusion tensor imaging shows us where restrictions to diffusion are and even the direction of them. And so red is right-left here in this image. And the blue fibers here are the corona radiata. They're going head-foot. And then green fibers run anterior-posterior in this image. So you can see where the white matter is and which direction it's running with diffusion tensor imaging. Okay, so let's talk a little bit more about these 2 measures and what they're telling us about the image. And so let's look first at that stiffness measure, the storage modulus, or just the stiffness, okay? And I said that, you know, these white matter fiber bundles are quite stiff. They're quite large and heavily myelinated and fibers, and so they are stiff. And we can look at the relative stiffness, and we can see, I show here in purple gray matter, and then in green white matter, and then red's corpus callosum, and blue's the corona radiata. And you can see here that all the white matter regions here are stiffer than the gray matter regions. And then still, the corpus callosum is stiffer than the other white matter regions. So really, it's these highly aligned fibers. These fibers are the rebar in the tissue. And so they're really providing a really strong mechanical component to the tissue. Okay, so that's sort of the stiffness measure. And I also want to point out a couple things here. It's quantitative. So we have quantitative imaging. We get real units, real measures. Unlike a typical MRI image, just has an intensity and it's arbitrary. You go to a different scanner, different sequence, you might get a different intensity, but we get real units out here, and you can see that we get highly repeatable measures. We'll talk more about that in a minute. So it's pretty easy to understand the stiffness. The other thing in the tissue is this loss modulus or viscosity. It's how much energy gets dissipated in these mechanical stimulation as you go through the tissue. And a great way to look at that is by looking at these white matter fiber tracts in the corona radiata. So in the corpus callosum, we have a lot of fiber tracts that are really tightly packed together, going across from one side of your brain to the other. They're all sort of packed tightly together, and they're headed in the same direction. And if you think about it, you know, as rebar and concrete, that becomes a very stiff structure. The whole thing is tied together. In the corona radiata, we have fibers coming up your brain, and we have fibers coming out that are intersecting those. And they intersect with each other, and they have some interaction. And so as you put these vibrations in, you can imagine that these fibers might interact with each other, and they have a little bit more degree of freedom to them as they go and interact. And that's what's going to show up as loss of this energy. It goes through the tissue. And it's why MRE is giving us more information about the tissue structure than just an image might give us. So we're actually able to see how maybe these different fibers are interacting with each other as they pass through. And so if you look at this, we have gray matter down here again, but if we look at the white matter structures, the corpus callosum has a really low viscosity here, a really low loss modulus. And that's because all these fibers are together. There's not many degrees of freedom for them to dissipate that energy, whereas the corona radiata have these intersecting fibers, and they allow a lot of degrees of freedom as the mechanical energy propagates through. So this is what's got us excited. We're measuring different aspects of this fiber structure. and the structure of the brain, and really not looking at maybe what we might normally see in the image, but looking at how the different parts of that that make up that tissue are interacting with each other. And it's giving us quantitative, repeatable information from there that we can then compare across people and populations. We show damping ratio later. I'm just going to put that up on the slide. So damping ratio is how we quantify this loss of energy. And so you can see here, the damping ratio is quite low for corpus callosum and quite high for corona radiata. And you'll see that damping ratio as one of our outcome measures later. In order to have so you make a new technique, you make a new measurement, you really want to know, can I detect changes in people or across populations? So one of the first things you want to do is make sure it's repeatable. And so we've run lots of repeatability studies over there. This is one of the first ones we ran quite a few years ago now. And we scanned one person lots of times over 2 months. And we saw that we had coefficients of variation of much less than 10%. And so in terms of those quantitative numbers we're getting out of the imaging, these are really reliable. We have sufficient SNR. Over the years, we've determined how to measure that SNR so that we know we're going to get a good data set before we go through all the trouble of inverting it, which takes a lot of computational power. So at the scanner, we can get a good idea as to whether we're going to get a good data set or not, or whether we need to repeat it. And you might need to repeat it for multiple reasons. We've had it where the tube was not hooked up to the pillow, so the vibrations were not happening at all. You would be surprised how easy that is to do. But then again, too, even just the position of the head on the pillow, if the vibrating pillow is off center a little bit, doesn't hit the right part, then you don't get as much energy transmitted through. So it's really important to be able to look at this and make sure you get reliable data at the scanner. So we have built this sequence up. I wanted to give you an idea of what the technology behind it is. And so sort of what my group did, we've told you a little bit about the method itself. But really, this method was enabled because my group has been trying to get really high resolution diffusion tensor imaging images of the brain and really trying to push the spatial resolution and the signal to noise ratio. So in MRI, as you get smaller and smaller voxels for more and more resolution, you get less and less signal. And at some point, you don't have enough signal to really make a distinction in the image. And so we've been developing these high resolution sequences but correcting a lot of problems. So if you're trying to measure diffusion on a very small scale or vibrations and displacements at the 5 micron level, the brain is pulsing from the heartbeat. People are moving a little bit as they're breathing, and you have to accommodate a lot of these other motions that might occur. So we've developed ways to measure those, correct them in the image, be able to provide very efficient acquisitions where we correct for a lot of other non-ideal conditions in the MRI. And from this, this is sort of where we started in this MRE, as we had this really great high resolution diffusion tensor scan where we can even start to see, I like to point out this here, you can really start to see fibers that were just sort of a mix in before. This is what's considered high resolution in MRI for DTI right now, and we've been able to go to 0.8. So we continue to advance this technique to make higher and higher spatial resolution maps of the mechanics, and we're hoping to roll those in. We're really excited now. Carl, just over this pandemic time, has upgraded their scanner to Prisma, same as ours, and so now we're able to run our best sequences in the clinic on these subjects, and we're really excited to get started with that. Once you get the data, I just also want to point out it's a huge computational challenge. So processing these images and then getting the mechanical inversions. We use a lot of Blue Waters time. I didn't put that slide here. I put more on the image reconstruction, but we use a lot of computational clusters on campus and Blue Waters time in order to invert this on a supercomputer to do the mechanical inversions. And that's one thing that as we go forward, we're hoping to figure out what is required in terms of mechanical models in order to be able to get the necessary clinically relevant information out of this. But to start with, we want to get the best we can and find good applications for it. So with that, I am going to turn it over to Graham.
Wonderful, thanks, Brad. And as Graham kind of cues up here, I just want to remind people, feel free to enter any of the questions that come up in the chat. We'll go through those, and then we'll also open up questions to the audience if they just want to ask them then at the end of Dr. Huesmann's presentation.
Thanks, Brad. It's been really exciting and interesting to work with the physicists over at the Beckman. My favorite thing is when they describe the brain as concrete and rebar. I think absolutely shows the difference between a biologist and a physicist. So I wanted to just give you a little bit of background on epilepsy as a setup for applications of MRE in this specific clinical population. So it's actually really common. There's 50 to 60 million people worldwide who have epilepsy, and about a third of those are resistant to any medications. Seizures are bad. Every time there's a seizure, there's a little bit of neuronal loss in the tissue. So the more seizures someone has, the more brain injury they have over time. So a delayed start to getting medication treatment means worse injury and therefore worse function over time. And then for the patients who are resistant to medications, you give meds, they still have seizures. The gold standard for them is surgery. And only about 5 to 50% of that population is ever even considered for surgery. And it's really due to a lack of access, which we're working on changing here at Carl with our epilepsy center. And we've hired a bunch of new epilepsy docs. And we have great partners with Dr. Mustafa and Dr. Olivero. So there's really, for simplistic purposes, you can break down epilepsy into two types. There's temporal, and then there's everything else. Temporal accounts for a third to a half, some people say up to 70% of all epilepsy. And a lot of those people start being controlled by one med, and then over the time, they lose their response to medications and they become resistant, and then they should be considered for surgery. And there's just a little picture of the brain showing the lobes, and then a ghost image of the hippocampus deep inside the brain. And the hippocampus is the part that's most prone to be the origin of seizures in temporal lobe epilepsy. And then the extratemporal seizure is usually due to a structural change in the brain, scar tissue, old blood, a brain tumor, congenital malformations in the brain. And about a third of that population is also resistant to medications and could be considered for a surgical option. And then what I didn't put here were all the different genetic epilepsies that usually present in the pediatric population. Another important group though. So treatment and surgical issues in terms of taking care of these patients. So the first problem, the first issue is that there's a delayed initiation of medication trial of on average 2 to 10 years. So the person has their first seizure, or they have something they think might be a seizure, and it takes a long time before they ever get a medication. And over that time, they're accumulating injury to the brain, and they're making it over time harder to treat. Along with that is a delayed surgical consideration. On average, that's 20 to 25 years from the onset of the first seizure. That's a big chunk of someone's life where they're losing function from the injury caused by the seizures. And so that delay increases risk of death. It decreases memory function if it's from the temporal lobe. There's a decreased ability to work, drive, raise a family, and be a productive member of society. And I'll go into this a little more detail, but MRE has the potential to identify these patients earlier. Why does it take 2 to 10 years for someone with temporal lobe epilepsy to even be considered to try a first medication? I think that partly has to do with the way temporal lobe epilepsy first presents. And for those of you that don't know, if it starts deep, like a little focal seizure, it does not look like a Hollywood fall down on the ground, hucka-bucka kind of seizure. It's literally just a feeling, like a deja vu feeling, or a feeling of anxiety, or a feeling out of body, or sometimes smelling things that aren't there. They're things that are subtle, they're short, they're 1 to 2 minutes, people can ignore them. Those start rarely, and then they become more common, and then they become daily. And as that seizure activity continues to happen, more of the brain is entrained and involved until they start with smelling something funny, and then they lose 30 seconds to a minute of time. They don't fall down, they don't look like they're having a seizure, but they're having a seizure enough to disrupt their awareness. And then as more brain gets involved, they can eventually have what's called a secondary generalization, where that seizure activity spreads to the whole brain. And then they have what people think of classically as a seizure, where they fall down and they have the tonic-clonic posturing. And that's usually when people present to a neurologist or an epileptologist for the first time. And unfortunately, that's a long time after the onset. But the question is, would anyone treat them if they presented and said, I'm having a lot of deja vu? They might if they came to an epileptologist. They might not if they went to a neurologist or to their primary care doctor. So MRE, and I'll show you some data on how sensitive it is, raises the possibility that we can have a measurement of changes in the brain that don't rely just on the history, so that we could actually identify these patients clearly early to begin treatment or begin timing of planning of surgery to give them the best quality of life possible. So in a classic, currently this is the gold standard, a surgical workup for a patient. To be a good candidate, they have to be under the age of 70. They have to be medication resistant, so have failed 2 medications, and the seizures that they're having have to be effective.
So Graham, you cut out.
Did he? Looks like he froze.
Into the EMU, which is the epilepsy monitoring unit, where, I'm sorry, can you guys still hear me?
Yeah, you're back now. Yep.
Okay, so they have to be, have been in the EMU, where we record patients in the hospital. EMU stands for Epilepsy Monitoring Unit. We bring them into the hospital and take them off their medications and get them to have seizures while they're being recorded on an EEG, so we can tell what kind of epilepsy they have, where it is, if it's one focus. We do a PET scan, which looks for decreased glucose uptake, which shows us that there has been injury to the brain from those seizures over time. We do an MRI, which shows flare signal or scar tissue in the part of the brain where we suspect seizures are coming from. We do neuropsych testing, which tests the best function of each lobe of the brain to see if the deficits correlate with lack of function in that part of the brain. And then we may do a WADA or an intracranial mapping, where we actually put electrodes into the brain to record from the seizure focus. But what do all these things have in common? They have in common the fact that there is injury. What these techniques that we have are measuring that the brain is already injured, that there's scar tissue formed, that it's not functioning correctly. So what do we need as a clinical tool going forward? Ideally, something that can show us the dysfunction before there's significant injury. And that's where I think MRE has potential. And one of the issues that comes up all the time in epilepsy surgical conference is when to do the surgery on these patients, especially temporal lobe, because you're going to be removing hippocampal tissue. And if you do it too early, you could induce some memory difficulty in them. You don't want your intervention to make them worse. And if you do it too late, it has the potential of injuring both hippocampi over time. And then it doesn't matter if you do the surgery, the memory is still going to be poor. There's a trial about this where they actually had trouble enrolling because people were afraid to take out hippocampus too early. But at 2 years after the early subjects had their hippocampus removed, they were a little bit worse in memory compared to the ones who did not have surgery. But at 5 years, the ones that had the early surgery were better than the ones who didn't have surgery, which I don't think it gives anyone great comfort to take out the hippocampal campus early, but it may help if we had a way of measuring that tissue and saying this tissue is at risk, and then really focusing in on timing the surgery to be the least injury from the intervention and the most benefit from when you stop the seizures. So this is just a slide to recap what I had just said. And it's really about what we're missing in the clinical workup right now is a technique to show that there is something different in the brain of these patients before the injury is present enough to be detected by the techniques we have now. Okay, so I'm going to try to go through this next part a little bit faster than I would, but I'm going to welcome you all to a surgical presentation of a patient that had surgery at Carl, and I'm going to present it as it would be presented at surgical conference. Usually Wael pipes in with something at this point, but all right. The patient is a 29-year-old right-handed female. No identified epilepsy risk factors, normal milestones of development. Starting as a child, she had feelings of fear, and that started at about age 10. And again, all it was feelings. So those could easily be ignored and may not even be talked about. Her first clinical seizure was at age 14. At that time, she had a normal head CT and a normal EEG. This is another problem. I'll just pause here for a second about EEG. The surface electrodes that we put on only see down about 1.5 centimeters. So the hippocampus, if you remember from that first structure, is really deep. If there's seizures happening really deep, you put the surface electrode on, it looks completely normal. So it's not unusual for someone who from the age 10 to 14 is having seizures, have their first clinical seizure, to have all of their imaging and testing look normal. And so she was not started on a medication. Her second seizure was at age 19, and that was blamed on another medication she was taking called Effexor. She was again not started on seizure medication or AED, anti-epileptic drug. Her third seizure was in her 20s when she was at home in Urbana on vacation from school. She was taken to Carl, and then an AED was started at that time. She did finish college at Arkansas, but due to decline in her ability to make memories and hold on to new information, she couldn't work in the field she studied in. When I met her, she was working as a maid in a hotel because she said if she had a seizure at work, she could just fall on the bed and she felt safe. There's a lot of stress in her marriage because she couldn't drive, difficulty doing tasks, and difficulty taking care of their young child at home. Got the MRI, and you can see to an epileptologist, this is a big signal, but you can see in her left hippocampus that there's a flare signal on the axial view, and on the coronal view, you can see it a little bit more clearly how bright that is in the hippocampus. On the PET scan, I always feel like it's looking through a lens with Vaseline looking at PET scans, but you can see there's decreased uptake of the glucose in the temporal pole and also into the mesial structures on the left side, suggesting that there's injury to the brain on that side. Put her in the EMU, and she had 10 seizures, all the same type of varying intensity, usually starting with an aura and then lip movements, difficulty speaking, and postictal crying. Some of those progressed to a secondary generalization while she was in the EMU. This is her EEG. Odd numbers are left, even numbers are right. So you can see this rhythmic higher voltage activity starting in the left frontotemporal region. And this is the beginning of her seizure. You can see that rhythmicity is just on the left, it's not on the right. And then as the seizure progressed, it becomes higher voltage, and then she goes into a secondary generalization where there's a lot of muscle artifact. So left seizures, left PET fits with that. Her neuropsych also fit with difficulty with more likely left temporal than right. And so the decision was made to proceed with surgery. So she got the surgery performed by Dr. Olivero over 5 years ago. And she's still seizure-free 5 years out. So before the surgery, she was having 2 to 4 seizures a week, and now she's seizure-free. And back working, back in school, they have a second child, her marriage is better. She's a very happy person and a happy, productive member of society. So in cases like hers, surgery arrests but does not restore memory function. So she hasn't really gotten back to where she was when she was a teenager, but she's, I think, backed enough that she can be a productive member of society. So what, with a case like this, she went from the age of 10 to the age of 30 with progressive epilepsy until she had a surgical intervention. And that was lack of acknowledging what's going on, lack of access to an epilepsy center, and I think also it shows a problem with the current gold standard of working patients up for epilepsy, and that we don't have a technique that can show us that there in fact is an injury and which side it's on early. So in order to, I'm going to go back one. In order to address that question, the beginning of the collaboration with Brad and Curtis and Hillary and Aaron was really the question, can we look specifically at hippocampus? And so, of course, grabbed a bunch of grad students and stuck them in the scanner to measure their brains, and got the elastography, and then did a hand-drawn map of the hippocampus, and then looked at both the volume and the damping ratio of the hippocampus, and we're pretty convinced that we could measure that specific tissue region with MRE specifically. How do I get rid of this thing? Okay, sorry. So those grad students, when we had them in the scanner, we also performed relational memory tasks on them, not in the scanner, but separately. So we had a measurement of how their memory function was, relational memory, which is a lot of hippocampal specific tasks, and then a measurement of their viscoelasticity from the elastogram. And when we look at volume compared to their spatial reconstruction or memory, there really isn't that much different. The correlation coefficient is only about 0.3, so it's a weak correlation. But when we looked at the damping ratio versus memory, the ones who performed better on the memory tasks also had a lower damping ratio. And you can see that there's a very strong correlation coefficient of negative 0.78, which shocked us, I have to say, that a technique could be so sensitive to detect differences between normal graduate students. And that has actually since been replicated, and in fact is a true correlation, which is kind of amazing to show how sensitive MRE as a tool. Some further housekeeping was also done to say, are we looking at changes that are specific to this type of memory? Because there's lots of different kinds of memory. There's short-term memory, executive function. So a task was chosen that separated out memory tasks or executive function that's more specific to the frontal lobe, orbital frontal cortex, and then separate tasks that were more hippocampus or relational memory. And then again, looked at the correlation coefficient between these two types and the elastogram from them. And you can see with hippocampus, with relational memory, there's again a good strong correlation coefficient, but it's not as strong with executive. Whereas in orbital frontal, it's not as strong with relational memory, but it is quite a bit stronger with executive or fluid intelligence measures, which I think is also strong evidence that the MRE is actually a very sensitive tool. It's also kind of amazing that this is in normal subjects. There were questions about what might be related or what might be causing the stiffness, and we're still working at. There's a lot of interesting questions about what it is we're measuring. But there's been a lot of work at the university over time looking at exercise and how exercise actually helps brain health. And so, again, looked at memory performance, but then also looked at cardiovascular fitness measures. And you can see that there is some correlation between fitness. The relative VO2 max improvement was correlated a little bit with increasing hippocampal stiffness, but not nearly as much as relational memory to the elastogram. So it does seem that we're measuring something that isn't just sort of blood flow related or vascular related. There's a separate sub-analysis of the data that was fascinating, where Anna Dougherty actually broke out by hand the differences between the subfields of the hippocampus of the subiculum CA1, 2, and CA3. Whoops. And she was able to, I think, fairly convincingly show in this paper that the region CA3 or the dentate gyrus is the region that changes the most. And that's where most of the viscoelasticity that we're measuring related to relational memory, which is, from a neuroscience point of view, kind of also fascinating, just because of how dynamic the dentate gyrus and CA3 are. Okay, so that's background. So what about epilepsy? So this is actually the MRE of the patient that I had mentioned in the surgical workup. And you can see her MRI showing the flare signal. This is the little mask we put over to measure hippocampus. And then you can see that there's a difference on each side between hippocampal stiffness from left to right. With just eyeballing it, you can see that the left hippocampus is stiffer than the right hippocampus. So we've enrolled, I think, about 14 subjects at this point who all had left mesial temporal lobe epilepsy, and then compared them to healthy controls. And one of the past gold standards of trying to increase sensitivity of understanding of the mesial temporal lobe epilepsy is, as that scar tissue forms in the hippocampus, it also loses volume and it shrinks over time. And so painstakingly measuring out the boundaries of the hippocampus, you can get a volume measure. And so we did that, and then both compared it to and added it to what we measured with MRE in that structure. And you can see with this ROC curve that volume measures in red are fairly helpful and predictive. That stiffness in blue is a little bit more sensitive. But if you add the two together, you get a really significant area under the curve addition of that. So we're not saying that MRE is better than everything else. We're saying MRE is a tool that, in addition to what we already have, could increase our sensitivity and therefore increase our clinical ability to diagnose. What this study that's still enrolling doesn't answer yet is, can we tell what those changes are early on? But we are scanning those patients as well. We just don't have enough to present anything yet. The data that we have from that last slide of those 14 subjects, we did quantify them in terms of severity based on a bunch of different metrics, how severe they were with their disease, because it does progress over time. And so we broke down the population into moderate disease versus severe disease. And when we do the sub-analysis of the data, you can see that the stiffness of the hippocampus does change over time. It gets a little bit stiffer when you go from moderate to severe compared to controls. An interesting potential in this early data is how the quote-unquote non-epileptic side. So in this patient, here's a picture of her MRI in the coronal plane again. The arrows on the left hippocampus. On the right hippocampus, which would be the non-epileptic side, we can see in this cluster of patients that it actually seems to get softer over time. So that raises the possibility that what we're looking for in early changes in temporal lobe epilepsy is actually a softening, and that the stiffening that occurs, the sclerosis that occurs, is the scar tissue that's forming after the injury has started. So that'll be a very interesting potential. This is my last slide. MRE works to detect mesial temporal sclerosis, and the signal seems to correlate with disease progression. We're still recruiting for that. And given how incredibly sensitive it is in normal subjects, we really do expect that we should be able to see changes early in the disease process. Because it's a structural measure, and I guess I should say physical measure of the tissue rather than structural, we should be able to see changes in every type of epilepsy, not just with mesial temporal sclerosis, but patients with small cortical dysplasias or small hemorrhages, and actually measure the changes in the brain tissue around them or in that structure that change over time. One hypothesis of how seizures develop and spread is called the kindling hypothesis. And it goes back to a very basic neuroscience concept of cells that fire together, wire together. So the more seizures happen, because those connections are strengthened every time it occurs. That actually is a structural change in the brain. So we may be able to actually see structural changes occurring as epilepsy is progressing if we measure people over time. Alternatively, we may be able to measure changes in the brain in response to treatment, because some patients, when you start them on medication, they stop having seizures and they have to stay on meds, but their epilepsy doesn't progress. So it has a huge amount of potential, again, not just for the most common type of epilepsy of temporal lobe, but for all types of epilepsy. And the ability to find these small cortical dysplasias is a high interest in the field of epilepsy, because about 40% of the patients who have medication-resistant epilepsy, suspected dysplasia, have a normal appearing MRI on 1.5T and 3T. I'm very excited about the 7T in order to put these patients into that scanner and try to find some of those really small lesions, but I'm even more excited to correlate that with MRE on the 3T and the 7T to see if we can find this concordance between what are called cryptogenic lesions on the 7T. So I had my little window thing on the side here closed, so I don't know if there are any questions, but that is all I have. So thank you.
Wonderful, very much. Thank you very much for both of you for just kind of giving that both the clinical picture and really the technology behind what you can really see with this. There are a number of questions in the chat. Maybe we'll start with those and then open it up for other verbal questions people might want to add. So one of the first questions came up when Professor Sutton was talking about kind of MRE and the resolution. And the question is, since tissues are mechanically coupled, is the spatial resolution of MRE really dependent on tissue type?
Yeah, so it's going to be interesting to determine what the spatial resolution limits are as we go forward and look at things. Right now, I would say our thoughts are, I need to put 4 or 5 pixels across the structure in order to be able to get a really reliable measurement of the mechanical properties, because we need to see how the waveform is changing in that structure. And so as the mechanical energy propagates through. So right now we would say that, but the hippocampal subfield work, I think, worked better than we thought it did, where we did get distinguishing features out of the individual subfields, and those are really pushing the resolution there. So, and different stiffnesses of different structures will affect the wavelengths in those tissues too. So it is possible that there is an interaction effect there between what we can see and what's there. We haven't had a whole lot of opportunities to look at that. We've done some phantom studies in the past, but once again, if you're not matching exactly the resolution requirements, then we do see that, as we violate the, say, 4 or 5 pixels across the structure, we're not completely losing sensitivity to the stiffness there. It's just that we start to, it's like a blurring process then on the actual stiffness measures, where they start to become closer to that of the tissue around them. So, so in the cases of the phantoms that we made, although the stiffness measure is wrong, it still shows as more stiff, than the surrounding tissues. So it'll be interesting to see, and probably application dependent And I think this is one thing for, another great thing for sort of, CI med students to think about too is, quantitative imaging is good and important and it's great and you can compare across systems too, but then you have to kind of be sensitive to what the clinical question is and what it's telling you too. And so, for example, as Graham showed, if more of a ratio between two measures is going to be the most sensitive detection, then maybe absolute quantification may not be as important as just signal noise ratio or making sure you don't have a whole lot of variability in those two measures. So yeah.
Okay. Yeah, thank you. Another question, technical question. So do you achieve resonance or a high Q in the brain tissue with MRE? Or is everything over damped at the frequencies that you're oscillating at?
Yeah. So we're not resonating people's heads. I don't know if Aaron Anderson is still on here though. We'll let him talk about Qs, but we're not hopefully not resonating people's heads and having them shake like that. But go ahead, Aaron. Yeah.
Yeah, so this is an overdamped system. We don't have any concern with actually hitting any sort of resonance. And to the point where we have to make sure that the amplitude's high enough and we pick a frequency of actuation that actually penetrates all the way through the brain down to the hippocampus. So in some cases, we try and go to higher frequencies because it is a viscoelastic material and has a frequency dependence. And in that case, we don't always get full penetration into the center of the brain. So we actually do have to play a little bit with frequencies and amplitudes to actually get that. So we're heavily damped in this case. Okay.
Good, thank you. Another question. So what kind of work has been done to validate your models?
Yeah, so it's interesting. Validation with MRE over the years, there's been a lot of efforts, and elastography exists across a lot of different modalities too. So even Steve here is doing, optical elastography and at a different spatial scale than we are, certainly too. So, but there is a frequency dependence of properties, and that can create some problems when you try to validate. So what are you going to validate against? One of the things that people originally were trying to do is indentation studies to get stiffness measures. So if you were to measure brain and then take it out and then do an indentation study on it and measure the mechanical properties, do you get the same response? And the answer is no, you don't. And there's a lot of reasons why. So there's frequency, there's the tissue. Once you take it out, the properties change. I will mention something along those lines that is an upcoming feature, if you will, of MRE that we're trying to figure out what the impact is on our measures right now. But people are starting to do functional MRE, where the reactive hyperemia that happens during brain activity, people are saying that they're seeing a change in MRE properties associated with that too. We're looking at it from the other perspective of how much of a variance can that have on an unintended variance on our measures that we're making in people when we're not controlling their caffeine intake or anything like that before they come in and have our study done. So, there's a lot of reasons why. So the right type of validation measure it's hard to come by. And that kind of goes back to my answer before too about the actual quantitative nature of things. It's reproducible, reliable. We get a number out. We were to do our scan and study on any number of imaging equipment, we're going to get the same numbers out. But are those actual stiffness measures of the tissue? They are for our study, but are they a property of the tissue? I think that's something left to be determined. So we can do phantom studies, and other things too, which we've done, and we get a high degree of reliability between those. But in the end, trying to compare that back to the properties of the actual brain, where it's very heterogeneous, it's very fine scale, and there's a lot of things going on, it becomes a challenging thing to actually say you have a validation.
Yeah, okay.
And how's that for avoiding the question completely?
Well, this next question maybe goes into a little bit depth there, but someone's asking, what are the limitations of MRE as an imaging modality? But in terms of, for older patients, do they have naturally a higher stiffness of their brain tissue? So does that impede or alter the imaging sensitivity?
Yeah, so limitations. So certainly stiffness, damping ratio, change as a function of age. We have some initial results on those things, as do other people. There's been a couple other groups that have looked at aging here too. We have an RO3 funded now. Hillary has an RO3 funded to run some older adults and really do a more careful study of how things are changing and how they're related to the memory performance measures that we're getting too, and whether those correlations are fixed across the lifespan or whether they vary.
Okay.
And so I think those are going to be some things. Limitations, I think, you know, we, as people, as their brain properties change and their geometry, even inside the brain and outside, change, we have to make sure we get adequate stimulation of the tissue that propagates through the tissue in a way that we get an adequate SNR, a signal noise ratio, across the brain to make these measurements.
Mhm.
And then we're not going to be able to measure really small structures with this until we can improve the resolution, which 7T is going to provide us some of that ability too. We're really excited to get cortical level things in 7T. So there are some limitations there. It looks like Graham has.
I was just going to add sort of the opposite answer of one of the assets of MRE is that you don't have to buy a $6 million piece of equipment in order to do MRE. You have to buy this resounding device, which then can be added to, in theory, and added to any scanner as long as the technical aspect of that is correct, as Aaron had mentioned and as Brad had just mentioned. So I don't think of it as being a limiting imaging modality. I think of it as being something that is added to an MRI sequence that you're already doing anyway.
Mm-hmm.
Yeah, and to carry that on too, I mentioned a couple of limitations, taking Graham's cue there a little further, that, you know, right now the way we do it is we are making the most sensitive, most careful measurement we possibly can. And that's at sparing no expense in terms of we're trying to get acquisition times, trying to keep them around 10 minutes or so, fine. But we have Prismas, we have the high performance MRI system that we're leveraging with our very special sequence with raw data pathways that include supercomputers. And so right now, that is a bit of a limitation. There's been a lot of places that want to collaborate, and we have other versions of our sequence that directly give images on the scanner, and it's not really giving the same type of information. So in order for us to do an experiment, it's 50 gigs of data from one of these scans. It's a half a week on a cluster to reconstruct it with GPUs. And then it's what Aaron, you know, most of a week or a couple of days with a couple hundred cores on a supercomputer to get the stiffness maps out. And about 15 people, which is why it's a really good team here too, required to go from end to end there. So we've automated a lot of that. And with the new automation tools that are available that can run on clusters and things like that, it's really coming together nicely that we can do this. But we do, you know, in order to scale out to being a routine clinical study, or in order to share out with other sites, we have to consider computational and data throughput issues, which are a big limitation right now. Yeah.
Sure. Certainly not real time, but there is, you know, with computing power every day, it's getting better. So yeah. Here's more of a clinical question. So any experience with middle fossa and cephalocele as a cause for epilepsy and changes on MRE?
Not with MRE yet. But we have been scanning patients with various brain injuries of different kinds. And that's actually been mostly driven by Dr. Olivero, who was one of the first adopters of this technique. So we have some interesting sort of one-offs. And we do now have a protocol in place, IRB approved, where we can start scanning more of these patients so that we don't have to have like a fully realized IRB for that specific thing in order to gather this data. So certainly it's going to be exciting to gather some of this information going forward.
Very good. Bill, I know you're on here. Any other follow on to your question there about other things that might be detectable with these methods? I think you're muted there. But.
No, I don't really have much to add. I just had that specific question because my cousin was diagnosed with a middle fossa encephalocele, and that was causing epilepsy, and it took a really long time to, diagnose him, I guess.
Yeah, no, interesting. So I had, I've been fascinated by your work, both of your work for a long time now, and just the stiffening that you observe in the hippocampus as it correlates to memory, just seems like, just a foundational discovery. And are you saying that stiffness then, the stiffness is because of the connections that then mean that somebody has improved memory because of that?
I think it's interesting because it's healthy, and it'll be interesting to get Graham's thoughts too. And I know we've talked about this, and probably our answers change every time, and so we'll see. But it's interesting. Healthy college age kids and having such a high correlation coefficient with memory is an amazing finding. There's nothing else I can do with the MRI scanner that can give me that type of predictive value at all. So it's explaining 50% of the variance in their performance on a memory task with the damping ratio of their hippocampus. And so we would like to know exactly what the underlying factors are. We have a couple studies going on in order to look at various components of that. And so hopefully these studies will allow us to see a little bit more about what's involved. I will just hint to one of them that we're writing grants for and have done some pilot data for. And that is that the great relationship you see there is in male subjects. In female subjects, the relationship is not quite as strong. And so I think that gives a hint as to some of the things that we're measuring with this technique too. And so we've designed some studies to go address that component, but then also, trying to understand more about, I think the fitness measures and how they relate in and other things. Are there systematic differences in across this that we're really sensitive to in this population, and right now we don't know what those are. So trying to look at it again. I like your question though about should we, for grad school, med school, whatever, students, I think one thing we should probably start doing, and we'll talk to Aaron about this later, is we should probably start monitoring our own elasticity declines in hippocampus very closely over the years here and see how that goes. So so
well, I know I raised that question because I got that question from some of my own students in my class when I presented some of your work on MRE, and so there was a lot of interest, right? And does that correlate to to performance in school and exams, right?
We assure anybody on this call that wants to participate in our future studies that the data is de-identified, and we will not be able to affiliate it back with you, so don't worry about it. But yeah, maybe people take that around and put it on their Safer at Illinois card, what their hippocampal damping ratio is. So.
Hippocampal stiffness. Very good. Very good. So I think maybe one more question here. How long is a typical scan time for MRE, and would this replace FLAIR imaging in the clinical workflow?
Yeah, I can answer the time really quick. So, as we're trying to get high resolution and trying to increase the sensitivity for a 2 millimeter isotropic scan, which is pretty good. And most of what you saw here are papers that have already gone through the publication process, which means they were acquired a little while back. And those were probably 2 millimeter for most of those. But the high resolution hippocampus, I think, went to 1.6, and now we're pushing for 1.25. The scan times are getting a little bit longer with those. And I showed a quick image in my part here with some of the times, but we're trying to keep it around 10 minutes to the top end of that. You have to put a pillow behind people's heads, and we're trying to make that a little bit easier when you put people in so that it doesn't take extra setup time too. But we could go anywhere from a couple to 5 minutes all the way up to 10 minutes to get data, depending on what it is we're trying to get. So as for replacing FLAIR, that one we'll let Graham speak to. Yep.
I don't think it would ever replace FLAIR. If it did, it would be decades, partly because it's a new technology, and that would have to go through many, many different iterations of clinical experience in order to replace something that's already tried and true. I understand the question because, FLAIR is looking at scar tissue, and in theory, MRA should see that better. I think it will be in addition to FLAIR once this starts to go mainstream. And in terms of it being not a long scan, which is good, maybe one thing to end on is when we had a patient not show up for a research scan, I hopped in the scanner just to see what it would feel like. And it sort of felt like I was resting my head on an electric toothbrush. So a nice little massage.
There you go. couple it with a neck massage, perhaps.
Right.
Very good. So I'd like to open up any questions that people had. Feel free to unmute yourself and ask them.
Yeah, this is Isam. A question to Graham, and this may not be related specifically to imaging, but you mentioned a patient that had hypoamygdalectomy. Are there any consequences neurologically or autonomic function after resecting the hippocampus amygdala on one side, or there's compensation?
There isn't. There actually isn't much consequence to that resection in patients who have advanced epilepsy on the side you're resecting. And that's probably due to the fact that a lot of the tissue in the resected area is already dead and has already lost a lot of connections. And they've already compensated for the loss by moving those functions over to the other side. And if the surgery is done correctly, which it always is, the insula isn't anywhere near the resection site. So you're not going to be causing any direct injury to autonomic functioning. It's an interesting question, though.
Thanks.
Another question popped up in the chat. So how does the variance observed between healthy controls compare to the variance between patients with MTS and healthy controls?
Yeah, that's a good question. In the healthy controls, we tried to match them for age, gender, handedness, and education with plus or minus 5 years in age. So in terms of variability, even across all the different ages we looked at, there was very little variability in the stiffness of the hippocampus. It's pretty consistent. It wasn't really enough data to say how the hippocampus changes over time. I think it'll be fascinating to see Hillary's aging data with that and then compare it to the healthy, normal, younger students like graduate students. The variability in the MTS, as I had shown, sort of does change with severity of disease. So as the disease gets more progressed, the hippocampus gets stiffer. So there's certainly significantly more variability within the hippocampal, within the disease population compared to the control. But obviously, it's only 14 subjects so far, so.
To be determined, yeah. Other questions?
I had one for Aaron, if he doesn't mind. Are you guys gating at all with the frequencies that you're doing and when the scans are acquired? Or is it just up to like some massive computational power to reconstruct the signals from the vibrations in the actual images?
So we don't gate with the heart. You can gate with the heart as a secondary actuation. So actually, instead of having the external vibration, you can use the pulsation of the brain, and then you gate it with the heart, and then you get it at about 1 hertz instead of 50 hertz in this case. And so that's looking more at how fluid flow moves through as like a porous material. So we actually think about it as a different material at that point. But then on top of that, basically, because we're highly tuned to the frequency we're acquiring to, we basically see anything else as noise. And so for the most part, we just say, oh, that we know, we just take the Fourier transform and say it's 50 hertz. So we just take the 50 hertz and there. And that's not where the computational difficulty comes from. That's the finite element part of it. So we kind of cheat when it comes to that part of it, is we just say the rest of it's noise, which obviously has its issues, but for the most part, it hasn't meant too much of a problem.
Gotcha. Yeah, that's an interesting thing, right? Because it sounds kind of similar to like what they do with geological surveys to study the core of the planet and with seismic activity. And kind of curious to see kind of how that propagates, and because obviously the brain's a lot squishier than the planet. So I was kind of curious to see if you'd stolen anything from US Geological or maybe some of the other physics guys that are, doing all the laser shaking and stuff like that.
Yeah, so actually Dartmouth, their group basically takes the same framework and they apply it to about 6 different problems. And one of those is related to surface waves. And they've also looked at using this for breast cancer detection with only having information at the surface. And so then they can take that and infer information at the center and look for lesions, for cancer lesions. So they've lots of different formulations. In our case, we benefit from having full 3D information, and we leverage that as much as we can.
Yeah,
Awesome. Very cool. Thank you so much.
Some of Aaron's thesis work kind of has another approach kind of like that too, is putting the stimulation in different regions. So instead of just a pillow behind the head, putting a pillow beside the head too, and then even looking at different frequencies and trying to mix that together to really figure out. So I think, yeah, there are a lot of crossovers with what people are doing with setting off the explosions and measuring the vibration. So yeah, that's good.
Good. You mentioned, how blood flow and those pulsations gives you kind of a 1 hertz stimulus. What about speech? Could you have people sing or talk, and then you can vary the frequency? Is that enough of a vibration to sense?
Yeah. So the Dartmouth group had trained people to hum at a certain frequency to be able to trigger that type of vibration too. I think it's a little harder to control the amplitudes going through there, and then we're acquiring images over a period of that 10 minutes where we're trying to sample the waveform at very specific points, and triggering off an ECG or something might allow you to synchronize and line up, but triggering off somebody humming is a little bit harder. So
yeah, interesting.
But yeah, if your goal, and it'll come back to the clinical application, I think, if your goal is to get some limited information about, the mechanical properties where you don't need so many time points and high resolution, you're probably going to be able to leverage some of these components and see how things go. Obviously, the low frequencies propagate through the head really well. And if you apply those externally, they're very uncomfortable. So having the internal humming and things like that is a good way to do that. But yeah, I think, as we get this tuned up and figure out what the clinical applications are, it'll be interesting to look and see what's the minimum requirement to get the information that you need out of the image.
Mm-hmm.
Right now, we're just trying to get it all. Yeah.
Yeah. So Zach
right,
has, oh, go ahead.
Yes. Yeah. So Steve, this is Kingley here. I'm a radiologist. So I can still remember the early days of MR when we actually scan anything with the limited technique that we have, and then develop chemical usages, right? And even now, right, it's hard to even understand how T1 and T2, right, relaxation times develop from basic properties.
Mm-hmm.
I see the same arguments here, right? MRE measures some composite characteristics, and we may not be ever able to sort out what contribute to the component measures that you're measuring. But it doesn't mean that it cannot be applied to a wide spectrum of diseases, right? So just like T2, T1 weighted imaging can be applied to a wide spectrum.
Yeah, I think that, I think you dropped out, King, but
diseases.
I think
and if we have enough time, you may find clinical utility in areas that you may not even suspect at this stage.
Yeah. And that's kind of, I think, the approach we're trying to take, but at the same time, we're trying to study the mechanisms. And so that's a little bit disheartening. At the same time, it's encouraging. And so, but yeah, I mean, what is quantitative imaging? Is it repeatable? And what information is coming out? And that's going to be the critical feature to identify. And that's not something we as engineers could ever hope to do on our own either. We have to understand what are the critical things to measure, and where is the useful information coming from, because that's really beauty in the eye of the beholder there for sure. Who's going to use that information? Who needs to make the decision? And we need to try to provide that and not get so focused on, completely understanding where it's coming from, which is hard.
Yeah, which is very hard, right? Even if you look at MR imaging, the numbers you're getting out of different machines are very different, right? So, and to go back to fundamentals and say, are you truly measuring T2 relaxation time? Are you truly measuring T1 relaxation time? None of that is really done, right, in routine clinical imaging, but it doesn't mean that it has no clinical value. In fact, we depend on it, right? Just looking at relative cellular intensities and so on, right? So a lot of it is actually how to squeeze clinical utility out of something that you may not understand 100%, right, mechanistically. So
I think Dr. Lee, you're basically raising an excellent point as to why we have this medical school. That the difference between it's an interesting scientific question, what is the underlying mechanism of this, versus, hey, there's a signal and it correlates with this, let's use it. And then, finding that bridge between the two. I'm sort of more in the, hey, let's use it camp. But putting on my neuroscience hat from my PhD, totally fascinated in trying to figure out what is changing in the brain, what is the underlying structural change that's causing these changes in epilepsy. Which gets back to that question from a while ago, which Brad had answered and hinted at. There's a difference between males and females. I think a lot of the hippocampal structural changes is actually due to synaptic density change. But there's more to come on that. I'm sorry I interrupted you, Dr. Lee.
Oh, no. I think that the two has no conflict, right? Developing clinical utility and understanding basic mechanisms can go hand in hand. But if you actually have to understand perfectly the mechanism before you apply clinically, we have very few clinical imaging, right? Techniques that will be satisfying all those requirements. So I would suspect that once it is clinically available widely, it will be applied to many, many different situations, and people will sort out what is the clinical utilities of it. I think the limitation now is that we do not have wide availability of the technique because of the computational requirements, right? So once I think that is overcome, just like the early diffusion imaging, it was tough technically, but once that is solved, you would be seeing people applying diffusion imaging in all kinds of ways, right? The same way MRE would be applied in many different situations once it is clinically viable.
I'll just jump in as the engineer. This is the tension, I think, in our collaborative relationship of me always saying my PhD was basically, this is how many different ways it's wrong. And Grandma said, yeah, but it's good enough. Let's go try it. And so I think this is sort of it's good to have that sort of balance, because otherwise we'll be stuck on the university side, just iterating instead of actually using it. So.
Yeah, it reminds me of the old days of how we developed MRI as a clinical tool, right? People were still trying to study what is the basis of T1 and T2 relaxation times. We're already imaging all kinds of stuff, right? And say, hey, I can see the difference between this and that on a T2 weighted image, right? And this is still most of what we do clinically. But fantastic work, though. I think I think it gives a totally different tool in a toolbox, right? For looking at diseases where if you just rely on diffusion, T1, T2 relaxation time, you may have a hard time sorting out.
Yeah, it's a whole different metric for measuring the brain that's never been available. The physical properties of it, the palpation of the brain.
Absolutely. So I think that's just another dimension, right, that we haven't explored. And there are many potential other dimensions there, right? That if we are smart enough to find ways to probe, we can develop fundamentally totally new techniques.
I think we had one last question. Zach, unfortunately, it looks like he stepped off, but he was asking if you've looked at the veteran population with TBI, or if that's in the plans.
Aaron,
yeah, it's in the works. Yeah. So the Ches Veteran Center on campus, which focuses on veteran health and in higher education, actually provided us with a seed grant to look at post-traumatic epilepsy. So, and then comparing why they develop epilepsy, whereas some of the people who have epilepsy, or sorry, have a TBI don't develop epilepsy. And so we're targeting that specifically at veterans. We'll take anybody with PTE, but we will be focusing on veterans specifically, because it's actually a problem that's been happening. The rate of PTE is basically held constant over the last 100 years, which is kind of incredible given how much warfighting has changed and advances in medicine.
Yeah. Wonderful. We'll pass that on. But thank you again. This has just really been enlightening, and it's just it's so wonderful to kind of see the synergy between the clinical and the technology to solve these problems, and also think about what's in store for the future. I mean, we look forward to that.
Great.
Thank you too for all those that have stuck with us till the end here. It's always a pleasure to have such a great discussion following these talks, and we look forward to many in the future. So thank you, everyone. Have a good